All right, so in this part of Chapter 7, we want to look at reactions from biological systems, and importantly, we want to look at the production of ATP, take a look at reaction spontaneity and when reactions happen, then look at enzyme-mediated reactions, and gain an introduction to pharmacology. So this first one we'll look at is ATP production. ATP is the currency of the cell that is used to do processes such as anabolic metabolism, or the building of our major macromolecules that we focused on in the last chapter. So making proteins, lipids, carbohydrates, DNA and RNA. It can be used for the movement of molecules into and out of the cell, or even within the cell. It's used for muscle contraction and neural activity. We saw the sodium-potassium ATPase pump being critical for the action potentials created in neurons. And it's used for much more inside the cell. All right, so let's take a look at our molecule of ATP. ATP is adenosine triphosphate, and this is a building block of DNA and RNA. This is the RNA ribose, the adenosine base, and it contains the 3 phosphate groups, 1, 2, and 3. So you can see it's the triphosphate. This molecule, when it is hydrolyzed, it makes the ADP molecule, or the adenosine diphosphate molecule. And so once it's hydrolyzed, it releases a lot of energy. And this energy is used to power neurons, do muscle contraction, and all of that. So you can see that last phosphate group has been cleaved off. Right? You only have 2 remaining in the diphosphate. So we use our food molecules to harvest electrons from, and this is going to provide the energy inside the mitochondria to be able to recreate our molecule of ATP by adding back that phosphate group. So one key feature of our ATP is that it has to be recycled. We use our body weight normally in ATP every day, but only a small amount of ATP even exists within our body. So we keep needing to recycle it between the ADP and the ATP to provide enough energy to power our bodies during the day. So this process takes place inside the mitochondria, which is noted as the powerhouse of the cell. It's the location where most of our ATP is recycled. So you can see the mitochondria contains 2 membranes, that inner one that is all wrinkly and the outer one that is maybe more circular or smooth. It also contains its own DNA. And so this suggests that mitochondria were once free-living bacteria that were engulfed by an early eukaryotic cell. And so when it was engulfed, instead of being eaten by that cell, it became a symbiont. So we think that this is from bacteria, that inner membrane from the bacteria, the outer membrane from the host when it engulfed it, and then it has its own mitochondrial or circular DNA that resembles bacteria. So our ATP production in the mitochondria is called oxidative phosphorylation. And so the oxidative part is meaning that electrons are pulled off of our food molecules. So remember, Leo the lion says, grr, if you lose electrons, you are oxidized. So if we take electrons from our food molecules, they become oxidized. That's the oxidative part. The phosphorylation part is the reaction where you're placing the phosphate group back on the ADP to recreate ATP. So the oxidative part is coming from our food, the phosphorylation part from placing the phosphate group back onto the ATP, and phosphate is often termed Pi. All right, so this is another diagram of our mitochondria, just a piece of it. looking at a close-up slice so that we can see the process of oxidative phosphorylation. So just looking, you've got the cytoplasm of the cell, you've got the outer membrane, you've got the inner membrane of the mitochondria. The space that's green is the intermembrane space, and the space that's brown is the matrix or the inside of the mitochondria. So in step 1, we are taking electrons from our food molecules, and they are going to be carried to proton pumps in the inner membrane of the mitochondria. So our electron from our food is going to go to these proton pumps that are shown in yellow. They transfer those electrons from each other, and then that energy from the electron transfer is used to pump the protons, the H pluses, into the inner membrane space. The electrons get transferred through that system, and they end up being reducing a molecule of oxygen. Those protons get moved from the matrix into the inner membrane space. This is important because it's building up a gradient of protons in the inner membrane space, which contains a lot of energy. So that's what's happening there. The electrons, as they move through the electron transport chain, will be donated to a molecule of oxygen. This is the oxygen that we breathe in every time we breathe, and it gets converted to that molecule of water. This is step 3 in this process. So the protons are accumulating in the intermembrane space, and they are like damming up water. They contain a lot of energy that when they can be released back into the matrix, they will rush into the matrix down their concentration gradient. So we said the electrons at step 3 get passed through all these pumps until they reach the molecule of oxygen. This is, again, the oxygen that you're breathing, and the oxygen is reduced. It's accepting those electrons. That's the GR part of Leo the Lion says GR. And it is reduced to a molecule of water. So what happens with the carbon dioxide that we exhale? Where does that come from? The carbon dioxide comes from the leftovers from our food molecules, right? We're going to be pulling the electrons and the protons off of our food molecules, leaving only carbon and oxygen, which gets released as carbon dioxide. So carbon dioxide is that waste from our food molecules that gets exhaled every time we're breathing. Pretty cool. So CO2 is exhaled from our food that we eat. And then in step 4, that proton gradient that is formed in the intermembrane space is going to be released through the ATP synthase protein shown in blue. The protons rush back into the matrix, and they actually physically turn that protein like a pepper grinder. And so when that mechanical force happens, it's enough to drive the production of ATP and add that phosphate back onto the ADP molecule. So you've essentially recycled your ATP from our food molecules, that the energy we pulled from them, and placed into the proton gradient in the intermembrane space. So this is both why we need to eat and why we breathe oxygen and exhale carbon dioxide. And this brings us to reaction spontaneity. When do reactions occur? So we've just seen with our molecule of ATP that energy is stored in chemical bonds. This is the energy of the attraction between the atoms involved in the chemical bonds, right? So this is known as bond energy. So if you try to pull apart two atoms that are bonded together, that's going to require energy to break those bonds because they're really attracted to each other and they want to stay together. So when you actually form the bonds, depending on how attracted they are to each other, this is the time when you can have energy release. So when we're assessing whether or not a reaction is going to proceed spontaneously, we need to determine which side of the equation that energy will be required or released. If you have to add energy to break the bonds on the reactant side, that one is not going to be spontaneous. But if you form more energy and you release more energy on the product side, those are the reactions that can happen spontaneously. So let's look at this in the context of the chemical reaction. So if you have to put energy into the reactant side of the equation, these reactions are called endergonic and do not happen spontaneously. They require energy to go forward. However, if energy is released on the product side, then these reactions are called exergonic reactions, and these are spontaneous because the bond formation is releasing energy, more energy than it took to break apart the bonds on the reactant side. So these reactions will be spontaneous, and they are called exergonic. So this energy of a reaction is called the Gibbs Free Energy. And if we look at changes in the Gibbs Free Energy, so that delta symbol or that triangle means change. And so if we are looking at the change between the products and the reactants of the reaction, it's possible to determine how much free energy is available to do useful work. If our change in energy or our delta G is negative, the reaction is spontaneous. So remember, our spontaneous reactions are exergonic, and energy is released on the product side. If our reaction is positive and the delta G is positive, the reaction is not spontaneous. This is the endergonic reaction, and this requires an input of energy into the reactant side of the equation. So energy is required, not spontaneous, endergonic reaction. There's one more type of reaction that can occur, and that's where our delta G is exactly 0. This is where the reaction is said to be in a state of equilibrium. This means that the forward reaction is happening at an equal rate as the reverse reaction, and there's no net gain or loss of reactants and products. It's in a state of equilibrium, equal amounts being produced and returned to the reactant state. So all of this that we've talked about thus far as the energy is concerned, is a consequence of the reactants and the products themselves. Enzymes have no bearing on the Gibbs free energy, right? This is an inherent property to those molecules and how tightly they're bonded together. So reactions are either spontaneous or not spontaneous. And even spontaneous reactions can take a very, very, very long time, even hundreds to thousands of years, to actually happen, but they will happen spontaneously at some point, right? It could just take a very, very long time. Okay, so if we're looking at a graph of Gibbs free energy, we can measure the change in the free energy between the reactants and the products over time. Right? So say our reactants require a certain amount of energy for that reaction to go to the product side, right? And then the products might be at a very different level of free energy. So here we could see that the difference between the reactants and the products is going to be below 0. Right? So we take the products minus the reactants, and this is equal to our delta G, or our change in free energy. So here, if we see that the products have a lower free energy than the reactants, our delta G value will be less than 0. We said that these reactions are spontaneous or exergonic. And this amount of energy difference will be released on the product side. However, if our reactants are much lower than our products, right, we will see that the product energy minus the reactant energy is going to be above zero, greater than zero. And in this case, we see that the reaction is not spontaneous. It can't go there. So one thing that we haven't talked about in the Gibbs Free Energy diagram is this middle bit in between the reactants and the products. And you'll see there's kind of like this hill in between these two that the reaction has to get over. And you'll see that you need energy to be put in to get the reactants to chemically react and change into the products. This here is the energy of activation. So it's like you have to push the ball up the hill to get it to roll down to the other side. This is where enzymes are going to have an effect, is in here. So that has nothing to do with the energy of the reactants or the energy of the products. Those are inherent properties to the chemicals themselves, but the path they take to get to the other side can be modified by an enzyme being there. So important to say again, an enzyme cannot change a non-spontaneous reaction into a spontaneous one. This is determined only by the amounts of energy required on the reactant side or on the product side being released. And if the energy of the products is higher than the energy of the reactants, energy needs to be added for that reaction to go forward, and the reaction is not spontaneous. Okay, so you're looking for that negative delta G is going to give us that spontaneous reaction. So enzymes can speed up the rate of a reaction because they lower the energy required to get to the transition state of the reaction. That's this middle place in between the reactants and the products where the chemistry reaction is actually occurring between the substrates to form the products. So without the enzyme, the hurdle to get over, or that mountain that this reaction has to climb, is greater. In the presence of the enzyme, the activation energy required to make the reaction go forward is lowered. So enzymes lower the activation energy. The activation energy area, this transition between the reactants and the products, is called the transition state of the reaction. During the transition state, unstable intermediate structures, like partial bonds that would be forming or breaking, happen during this period of time. And that's why you need this hill of energy to get those bonds to break and then form the new ones that are going to be in the products. So several factors can alter reaction rates of enzymes that are catalyzed by reactions. These include the concentration of the substrate, the concentration of the enzyme, the temperature, and the pH. And this is what our lab focuses on this week as well, is looking at all of these different factors that affect enzyme function. So in the presence of a given amount of enzyme, the rate of the enzymatic reaction increases as the substrate concentration increases until a limiting rate is reached. So if you increase the substrate concentration and we have the same amount of enzyme, so enzyme concentration is constant. You can keep increasing the substrate concentration and my reaction rate will be faster and faster and faster until some point, right? And then I can keep adding more substrate, and I can't get the reaction to go any faster, right? So this is my reaction rate. You can see that's increasing. Rate is just the formation of the product over time, right? And that can be measured in seconds or minutes, right? So this is going faster and faster and faster until you reach a maximum rate, right? So there will be some maximum rate. Right, and that makes sense because if your enzyme concentration is constant, eventually you're going to fill up all the enzymes, and then the substrate has to form a queue for the enzyme to be able to work on it. You know, it's got to get in line and wait until the products get kicked out of it, and then the enzyme can be used again. That's another cool thing about enzymes. They are not used up in the enzymatic reaction. They are just the worker, right, that is going to help mediate the reaction so that it can go faster. It's going to bind to the substrates, helps orient them so chemistry can happen, and then you form the products and release the products from the enzyme. But the enzyme is not used up in the process. You can reuse the enzyme. All right, but that means that you are limited in the reaction rate by how much enzyme is present. That makes sense. All right. If you increase the enzyme concentration and you have a set amount of substrate, you would see the same thing happen. Initially, you're going to see the reaction rate increase, 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 and increase. If you have no limits on your substrate concentration, it would continue to increase in this linear way. However, if you had a fixed amount of substrate, you would see that this line would then flatten, and that would just indicate that you have no more substrate present that could be converted into the products at that point. You've used up all the substrate. Okay, so that's how things change with substrate concentration increasing versus enzyme concentration increasing. And in this one, there is no limit on the substrate. Okay, so you can change the enzyme concentration, you can change the substrate concentration, and we've seen how those affect reaction rates. But we've also talked about the process of denaturation and that proteins are pretty sensitive to their environments. We said if you change the temperature, like if you're running a fever, you can start to denature the protein or melt the intermolecular forces that keep them in the correct three-dimensional structure. So most proteins have a temperature value where they work optimally. And for our proteins, this is going to be at about 37 degrees Celsius. That is our normal body temperature. And you can see that once we get past that and we keep increasing the temperature, you have denaturation of the protein at a high temperature, right? Or unfolding, and the protein is not going to work well anymore. However, if you go down in the temperature, you also see that you have less activity of the protein. This can be due to the kinetic movement of the protein. Like, I don't know if you've ever tried to do something when your hands are really cold or like almost like numb, right? Like, so if you're outside your car trying to put on chains and you don't have any gloves on and it's snowy outside, it gets really hard to even feel where the chains are and your hands slip and it takes you a long time to do it. Whereas if you just did it on a regular day when it's warm outside, you could do it really fast. That is partly like kinetic movement, right? It slows down when things are cold. That happens with your proteins too, right? Like the movement of the proteins is going to be slower. So kinetic movement slows as you reduce temperatures, and this causes protein reaction rates to also go down. So proteins, again, have this optimal window where they're going to work, where they're folded correctly, and they can move around optimally. This is the same for the pH range. Think about what's happening with the pH. If we decrease pH, right, so neutral is at about a pH of 7, That's where most of our proteins are going to work well because our body pH is typically neutral or very close to it. Maybe 6.8 to 7.2 is our range. And you can see that there's a pH window where our proteins will also act optimally. If we increase pH, we are increasing concentration of OH minus. These are charged molecules, right? That oxygen is carrying a full charge on it, right? And that's going to make our proton that's attached to it, or the hydrogen that's attached to it, become slightly positive as well. But we have a full negative charge floating around. That can cause a disruption of protein structure as well, because that's going to also interfere with the intermolecular forces, because this can get in there and form salt bridges or do other things with the protein and cause it to misfold. So you get misfolding when you shift the pH. And this can also happen when you're increasing, right, the proton concentration, or the H+. Now you have a full positive charge floating around there, and you also get misfolding when you go in this direction too. So protein denaturation will occur when you move off of your optimal pH levels. And this is due to the changing of protons or hydroxide ions in the solution. In this last section, we want to introduce you to pharmacology, or the branch of medicine concerned with the uses, modes, and mechanisms of drug molecules. The term mechanism of action, or MOA, refers to the specific biochemical interactions through which a drug substance produces its pharmacological effects. So drugs can act in many different ways on the body. So they could bind to cell receptors at the plasma membrane, right? So they could mimic a normal function and activate a receptor, or they could block the activity of a receptor by binding to it. They could cause increased protein expression, or they could repress protein expression as well once they get inside the cell. And this could be at the level of the protein. They could just bind to the protein and cause it to become more active, or cause it to become less active. They could also bind directly to the DNA in the nucleus, and they can cause gene expression or cause gene repression, where they block the expression of specific genes and then therefore block or reduce protein levels. They could target specific enzymes inside the cell and block their activity so that they can no longer make their products and their intermediates. And they can also alter cell receptors and signaling molecules so that they interfere with the pathways of signaling going on inside the body. And sometimes these will cause the activation of cell death pathways called apoptosis. And this is a programmed cell death that the cell can go into if it feels like it's under serious attack. So there's two major types of drug molecules that you can take. The first are called agonists. Agonists will occupy the normal substrate or ligand binding site, and they will produce a similar conformational change in the protein that leads to the enzyme or receptor activation. So agonists do the same function, essentially, as the natural ligand, whereas antagonists are going to block the activity. They can bind to the active site of the receptor or an enzyme, but they don't produce the conformational change, and they prevent the action of the normal substrate or ligand. So agonists or antagonists. So there are also two concepts that you need to understand a little bit about to understand how drugs work. First, the affinity refers to how well the compound will bind to the receptor, where drugs with higher affinity will exert greater or more long-lasting effects. They bind tighter if they have higher affinity. This is different from efficacy. Efficacy, on the other hand, is a measure of how well the compound activates the receptor. Strong agonists will also have high efficacy, whereas if you have a strong antagonist, it will have low efficacy and inhibit cellular pathways. So good antagonists will have high affinity but low efficacy, versus good agonists will have both high affinity and high efficacy. So let's just talk about antagonists a little bit further, as these can be divided into 3 subcategories based on their mechanism of inhibition. These include competitive inhibitors, non-competitive inhibitors, and irreversible inhibitors. So if we have normal receptor binding, this is the normal ligand that normally would bind and activate this enzyme or a receptor inside the body, right? So maybe this is dopamine inside the brain. It's going to activate the neurotransmitter in there. If we have a molecule that can block that activity, this is our drug molecule. It can also fit into and bind to the active site of our enzyme, and now it's blocking directly the activity of the normal ligand because it's competing for the binding site, right? So this is competitive inhibition. It is directly competing for the same binding site as the normal ligand. Competitive inhibitors can be overcome by adding additional substrate, right? If you have more of the natural ligand around, this thing is coming on and off and on and off. It's bouncing in and out of the active site binding. If you increase your concentration of this will take over and be able to bind more often to its normal enzyme, and the reaction will occur. So that's an important feature of a competitive inhibitor. It can be overcome if you add more substrate. A non-competitive inhibitor will bind to a distant site on the enzyme or receptor, and it will induce a conformational change in the protein that prevents the normal substrate from binding to it. So this type of binding is known as allosteric binding. Allosteric means away from the active site, right? So this would be the allosteric site here, away from the active site, which our normal ligand is binding to here. That's the active site. So when a compound binds to an allosteric binding site and causes a conformational change in the protein, this is called non-competitive inhibition. It's not competing for the active site. It changes the active site. And under this situation, you cannot overcome the inhibition by adding more normal substrate. However, the drug target will not be permanently altered. So this is still popping on and off, right? And so as long as you have enough concentration of the inhibitor, you're blocking the activity of the enzyme, but this is not a permanent binding to our enzyme. It's popping on and off. And so ultimately, you will metabolize the drug and it will go away from the system. And once that happens, your normal ligand can then bind again and you will get reactivation of that pathway. That's like the normal half-life of the drug. It's going to get metabolized in the body and processed and removed. Okay, well, how about irreversible inhibitors? In the case of irreversible inhibitors, they will bind to the enzyme and they will covalently form bonds with your enzyme. And if they form covalent bonds, rather than just interacting with intermolecular forces, these are tight, right? These cannot be broken, right? And so they are irreversible. So this protein can no longer be reactivated and function in its normal way or its normal pathway, right? So this is permanent. The only way to get rid of this, right, is to remake the entire protein. And oftentimes, there's no time to do that, and this might be too deleterious for the cell, and the cell might go into apoptosis or programmed cell death, right? For a unicellular organism like a bacteria, this would kill the bacteria. And so we would want to do that if we are using antibiotics to kill a bacteria that's harming us. So some of our antibiotics are irreversible inhibitors like penicillin. And that is where I will leave you for chapter 7. And now you should be able to complete your homework for this week.